Thank you, Sharon and Erin, for inviting me to talk and to chair this session today. I'm very glad to be here. I'm very glad to see so many people in the audience. As a clinician scientist, it's important that we get feedback and know that the work that we do is actually appreciated. Sometimes universities don't appreciate the work we do, so it's good to see patients appreciate what we do. I'm tasked now to give you an overview of the sort of research that's going on related to retinal diseases, retinal blinding diseases, with a particular focus on what's happening in Canada. That's, as you can imagine, that's quite a big subject. I could be here for a week talking about that. So since I only have half an hour, I'm going to have to cut it down. And I'm going to mainly focus on the sort of work that is one stage away from clinical practice. I'm not going to talk so much about the basic science, the basic concepts that are being studied, but more the translational research, where ideas have already come forward as particularly good, but now we need to show that they actually work in humans and then move that work on to being available to everyone and through their regular ophthalmologist. Now, going from a basic concept to routine clinical practice, it sounds pretty straightforward. So, for instance, if I had a treatment that worked now, right now, a brand new treatment that absolutely worked, it could take 10 years before you actually saw that treatment. And that's because there are lots of regulatory things that you have to go through before you take an idea and start giving it to patients. There are very good reasons for that. And the two main reasons, well, the first thing is it's illegal and, you can't break the law, so you have to go through the regulatory bodies. But the regulatory bodies are there for a good reason. They're there to ensure two things. One is that whatever the new therapeutics are, that they're safe. That is the first priority. And the second priority, and this is particularly true with emerging treatments for eye diseases, and that is that not only does that treatment have to work, it has to work well enough. If I have a treatment that would make people taller, for instance, I would be a millionaire overnight, because lots of people want to be taller, lots and lots. But if I had a treatment that was only going to make you taller by 1/4 of a centimetre, I'm not going to make very much money out of that. And sometimes new therapeutics can be a bit like that. They are working, but they don't work very well. And that's a problem when you move from model systems into human beings. Sometimes you see something working very well in a model system, but not very well in a human. The interesting thing is that sometimes it can be vice versa. It can work a lot better in humans. Some of you, especially those with age-related macular degeneration, will have heard of the anti-VEGF treatments like Lucentis and Avastin. They're an example of a drug that didn't work that well in model systems, but work phenomenally well in human beings. So these are the sort of things that you have to take into account when you move new research ideas into clinical practice. When it comes to genetic retinal diseases like age-related macular degeneration, Usher syndrome, retinitis pigmentosa, Stargardt's disease, all as a group very common causes of blindness in the Western world. Research into new treatments is very, very active. In fact, you can be reassured that of all the different parts of ophthalmology that are under research investigation, retinal diseases are the most intensively researched. And there's a good reason for that, and that's that they are so common and they are so difficult to try and treat. You could go back a decade or so, and for the vast majority of retinal diseases, not only were there no treatments, there weren't even very much in the way of ideas of how possibly to treat them. So we've seen a lot of progress in the last decade or so, and the Foundation Fighting Blindness has had a big part in pushing forward the innovations that are now emerging. essentially, we're talking about 4 areas of research. There is gene therapy, new pharmaceuticals, electronic retina and electronic devices, and stem cell research. And I thought I'd give you a rough idea of what's happening in each one of those 4 areas. When you talk about genetic diseases, the first thing that always comes to mind is gene therapy. And certainly for retinal diseases, a lot of work is being done in gene therapy. Lots of basic science has been done. There's been a lot of concern about safety with gene therapy, but I think those concerns have probably not become apparent in preclinical studies and in the early clinical studies that are ongoing. So safety isn't emerging as a big issue. In Canada, we're lucky to have 2 very prominent gene therapy experts in Robert Mulday here in Vancouver, who is particularly interested in gene therapy in preclinical trials for retinitis pigmentosa, but also for a common blinding disease in young boys called X-linked retinoschisis. So he's particularly interested in those areas. And we also have Ian MacDonald up in Edmonton, where I'm sure it may be raining here, but I'm sure it's snowing up in Edmonton and is probably minus 30. So we're much better here in Vancouver. But Professor MacDonald is leading a large clinical trial for choroideremia. This is an X-linked form of retinal degeneration that has a lot of similarities to retinitis pigmentosa. The clinical trials that are going on in choroideremia and the clinical trials that you might have heard of that are underway in the US and in the UK are still at a very early stage. Clinical trials conventionally are chopped up into 3 parts, and a research group has to go through each part. They're called Phase 1, Phase 2, and Phase 3 studies. Phase 1 studies are only supposed to be about safety, only about safety. Phase 2 studies are larger studies that involve more patients, and this is the time that you begin to look at whether there is a therapeutic effect. Phase 3 studies are the larger studies that usually will involve hundreds to thousands of patients, will probably be done at multiple sites, and that is the Phase 3 study is where you can actually work out how effective a therapeutic is. And all new therapeutics are supposed to go through these 3 stages. Gene therapy for retinitis pigmentosa and for Stargardt's disease is still very much planted in the Phase 1 stage. It's a safety study. The concerns for gene therapy in general are that it's thought that the virus vectors that are carried by, that are carrying the new genes can induce tumours or can induce immune reaction. Both, as I say, both of those things are not looking to be a particular problem. So gene therapy is looking very promising. I don't think, though, that gene therapy will be for every patient with retinal disease. And there's one good reason for that, and that is that a gene therapy approach is likely to inhibit disease rather than reverse it. So it's probably something where you can pick up disease very early and do the gene therapy at an early stage before serious vision loss has actually happened. But for a lot of retinal diseases, a lot of vision loss has already occurred when a patient presents. So for them, gene therapy probably isn't the approach. But certainly for things like early retinitis pigmentosa, choroideremia with Professor MacDonald, there's certainly a lot of value in this. One of the things that I used to see a lot in the popular press in the past was this idea of a magic bullet. The magic bullet, one thing, do everything. I think in vision for retinal diseases, there is not going to be a magic bullet. There are going to be magic bullets. There are going to be lots of them. And for a particular disease, and perhaps even for a particular stage of a disease, there'll be different things. And gene therapy will find a place in to treat one type of disease at one particular stage. Pharmaceuticals are something that I think that everybody here should be particularly interested in. Of the 4 approaches that I'm going to summarize, pharmaceuticals are probably the most likely to be seen in clinical practice in the near future. And that's because, to some extent, developing new therapeutic molecules is easier than the other approaches, because there is already a pharmaceutical industry to take new ideas and have the machinery to push it through the preclinical and clinical trials to clinical practice. Also, with all the research that's going on and the understanding of the molecules involved in retinal disease, every time a new molecule is found, a new idea for a treatment emerges. So there are very many clinical trials testing new pharmaceuticals, particularly in diseases like age-related macular degeneration. Here in Canada, Canada, in a lot of ways, is leading new pharmaceuticals in retinal degeneration. Later on today, you'll hear from Orson Moritz, who's going to talk about his work using sodium valproate to treat retinitis pigmentosa. But also, in addition to that, we have over in Ottawa, Catherine Safidis, who is working on a molecule called XIAP, which is an X-linked inhibitor of apoptosis. This is a molecule that stops cells from dying. So an end point for a lot of retinal degenerations is that the cell tries to compensate for whatever genetic abnormality it has, but there comes a time when it can't cope with that anymore, so the cell dies. And that's when you then see vision loss. In Ottawa, they've developed a molecule that seems to be able to protect the cell from that final death signal. And this is something that is particularly useful because it will be applicable to retinal degeneration despite the cause. So a gene therapy approach, for instance, is very specific. If you have a gene therapy for retinitis pigmentosa, it probably isn't going to work for choroideremia or for Stargardt's disease, for instance. But the XIAP molecule might work for all these. So this is a particularly exciting area and is an area which is already going through towards the end stage of preclinical work and will probably be starting clinical trials very soon. We also have in Montreal Robert Koenigkoop, who is working with a Canadian company to develop retinoid drugs to treat the earliest stages of retinitis pigmentosa, a particular version of RP called Leber's congenital amaurosis, so a childhood blinding disease. Now, these are interesting molecules that target the biochemistry of the disease rather than the gene. So at a later stage, are more widely applicable. There is some potential there to actually reverse damage with these drugs that Robert is using. And these molecules are already in early clinical trials. Lastly, studies that are particularly of interest to AMD patients, and that is the antioxidants that have been developed by Bausch & Lomb, a cocktail of molecules that inhibit progression of the disease. These molecules have been around for a few years now, but Bausch & Lomb and researchers in Canada, and particularly in the US, are now refining that cocktail to make it more effective. So quite a range of things are being done in pharmaceuticals, and these things are already starting to appear in clinical practice. Electronic retina and electronic devices are exciting. If Steven Spielberg was going to write a film on treatments for retinal disease, it would be electronic retina. That's what it would be. So these are electronic devices that replace the retina. Very exciting research. Still, I think, at a very early stage. A company called Second Sight has set up a clinical trial, and I think it's mainly based in London and Ontario, with the Argus 2. So this is an implant to put into the eye. The sort of vision that you get with electronic retina, I think we're not going to see electronic retina restore sight completely. I think we're going to see limited restoration of sight, but still sight that could be useful. Useful for reading, perhaps even for driving. But I'm not very hopeful that the technology can develop vision. So, for instance, you'd be able to return good colour perception, or become a sharpshooter in the army. That sort of vision. I don't think that they have that potential, but certainly very useful vision for driving, for employment, for reading, and huge amounts of money are being put into electronic retina. Major research projects, as I say, here in London and Ontario. Lots of projects in the US. For some reason, electronic retina appeals to researchers in Germany. And there is huge government funding for electronic retina in Germany, and to some extent also in Australia. So lots of movement in that, and I'm sure you'll be seeing lots of electronic devices for sale and on offer in the very near future. One particular area, and the last one I'm going to mention, is stem cells. Again, stem cells have the aura of almost science fiction, but there is a huge amount of science fact to go along with the science fiction concept of stem cells. Stem cells work. Stem cells are the future of medicine. There is no doubt about it. There is no doubt that in the future, stem cells will be a therapy for genetic retinal diseases. The only question to ask is when? Is it going to be sooner or later? There is no doubt about the technology. It will work. It's just a matter of when. We have a leading researcher in the world involved in stem cells in Canada. Valerie Wallace is a star of stem cell research around the world, and we are very lucky in Canada to have somebody so dedicated to focusing her stem cell research on the treatment of retinal degenerations. And we will see clinical trials of stem cells in retinal degeneration within the next few years here in Canada, hopefully here in Vancouver in the very near future. You'll be aware of the trials that are already going on in the US with Steve Schwartz, and also in the UK with Jim Bainbridge and Robin Alley, looking at patients with retinitis pigmentosa and with Stargardt's disease. These, again, are phase 1 studies, but the phase 1 studies have proven to be very successful, and both research groups are looking to move into stage 2 to start to show how effective these cells are. So I think we've got a very packed day today, so I'm going to stop there to give everybody else the opportunity to speak. But I'm willing to answer any questions later on in the day, and I hope you all enjoy the rest of the talks. Thank you.